Live, 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 live. We're live. We finally went live. Justin's awake and we finally went live.
I wouldn't go that far, but okay.
Welcome everyone to the live TWIS broadcast. We are here. We hope you are here. I got green check marks all across my networks, which means to me that we are streaming. Welcome, everyone. Oh, I just got the It's Live from Identity 4 in our chat room. Haha, Kevin Unique is waiting on TWIS and Godot. We're here. Shoe Brew says we're live 5x5. Fantastic. Well, we are just about ready to start our show after this wonderful day. Today was a wonderful day. I'm full of happiness. And those of you who are joining us right now, thank you for being with us tonight, because I know there are other things, celebratory things that people could be doing and watching. And if you've chosen to spend your time with us, we do appreciate it. It's a good day for science. It is, Gaurav. Indeed. All right, Justin, are you ready to start a show?
Yeah.
Were you able to get your fonts, your text at a size that you can read?
No, but I kind of know what's there. It'll be fine.
It's all right.
Yarps.
Yarps. I feel like we're on a little diagonal slide. No, I gotta do the other way. There we go. We're ready to go.
Okay.
The beauty of non-mirrored video.
That's right. Very.
Yeah. Like that. Oh, look, there's the top of my head. More head toppings. All right, you ready, Justin? You ready for this?
Absolutely.
This is how our show always starts, with a little hemming and hawing and putting it together, and we will begin the show. Do I seem quiet? I haven't done anything to my audio levels. That's so weird. Some weeks a little quieter than others. I turned myself up just a little. Just a little bit. Let's see if that works a little bit better. 1, 2, 3. We ready to go? We are ready to start the show. Yeah. Okay, let's do it. Beginning the program of our live TWIS broadcast. What you see here in front of you today may not be what makes it into the podcast, so enjoy what you see. In 3, 2, this is TWIS. This Week in Science, episode number 808, recorded Wednesday, January 20th, 2021. Science loves palindromes. Hey everybody, I'm Dr. Kiki, and tonight on the show we will fill your head with old cloacas, protection, and hope. But first...
Disclaimer. We are living in interesting times. Well, this has always been true to the people alive in some other times. This day, in this age, will be of especially key interest to future historians. Future historians who will be tasked with figuring out how our societies got to where we will be at the time when future historians are tasked with figuring out our present future foundations, or their future present foundations. It gets confusing. Anyway, we will look back someday on this time and try to figure out what happened. Will this be the age where policy discussions are conducted by people with knowledge, interest, and commitment to making a better world? Will this be another era of war and destruction? Will the actions taken by humans of today lead us to a rational world of reason, technological innovation, and scientific discovery? Or will we continue to see truth, facts, and knowledge further discarded in favor of the fervently low information minded, the faithfully foul-mouthed, feckless mobs with upper-decker mentalities? Will the future historians cringe at our next 50 years? Or will they rejoice, us as pioneers of a future they hold dear? The answer can be found in our recent past where it seems anything can happen. All the possibilities are still laid out in front of us. We are living in interesting times, and nowhere is that made more clear than right here on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science, Blair. Welcome to
This
this.
was me not hitting the button fast enough.
Welcome to this.
Good science. Yeah, good science to you, Justin, Blair, and everyone out there. I am just, I guess, excited to be here tonight on this auspicious eve of palindromes.
Huh?
Yes! 1/20/2021 is a palindrome, forward and backward. Our episode 808 is also a palindrome, same forward and backward. It's like the palindromic day.
What does it all mean?
What does it
Nothing.
mean?
No, literally, what does the word palindrome mean?
We could go into the etymology of the word, but really it's
good.
something that is the same forward and backwards. But if you listen to this show backwards, it will not be the same. So let's get started from the front and move to the back, to the end. It's the beginning of another great episode of science. We've got all sorts of great science news today. I have stories about the importance of water, cats and mosquitoes, and lots of AI learning. It's going to be great. Justin, what do you have?
I have fungal farming ants, hidden brain functions, pulsed laser technology, and a duo of twin studies, or a twin study of duo twin. It's not just two studies. Well, there's at least two studies, but they are on twins and the microbiome, sussing out allergies, which I think is good.
All right, suss it out. I like sussing out allergies. Give twins allergies. No, we'll find out what this story is about later. Blair, what is in the animal corner?
Oh, I have bad news about marine protected areas. Sorry. I also have rotting food. I have hunting eels. And I have a dinosaur butt with poop still in it.
That is a find. That is
Uh-huh.
something
Wow.
special.
It is a paleontological gold mine that we will discuss later.
Wow. Oh, I hope we do. We will dig into it
Yes.
later.
Good, 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 good.
Aha. But for now, for those of you who are not yet subscribed, you can find This Week in Science at TWIS.org, our website where we have episodes and show notes. We are also on YouTube, Facebook, and on Twitch live streaming. YouTube and Facebook, look for This Week in Science. On Twitch, look for TWIS Science, TWIS CIENCE. And you can look for This Week in Science anywhere podcasts are found, to look for TWIS as a podcast and subscribe. Let's dig into that science. Are we ready to do that?
Let's do it.
Let's do it. I think we're ready. Okay. I want to start us off with hope, because today is such a hopeful, auspicious, like I said, palindromic day. But beyond that, today was the day that the 46th President of the United States of America was inaugurated. And last Friday, President Biden, he wasn't yet president to
Elect.
be
Yeah.
Biden, yes. He announced his choice for science advisor and head of the office, the White House Office of Science and Technology Policy. This person is Dr. Eric Lander. Dr. Lander was on Obama's Presidential Council of Advisors on Science and Technology, and he co-led the Human Genome Project, which gave us the first sequencing of the human genome, and MIT's Broad Institute, which has been in competition for years for CRISPR technology against UC Berkeley. He is a firebrand of a scientific leader. a bit controversial in some areas. There are questions as to whether he knew Epstein in any way, shape, or form, considering he is in the same echelon of scientists that Epstein gave a lot of money to. But he has so far not had many links in that direction and has just shown an amazing ability to lead, to bring teams together to get science done. So this is exciting. In addition, his position has been elevated to the cabinet level for the first time in history. This puts science at the table, in the room, during important policy discussions. So for science, very excited. Hope. Additionally, Biden's scientific administrative team was announced last Friday, and it consists of a diverse array of well-qualified individuals, including science historian Alondra Nelson, Francis Arnold and Maria Zuber, Francis Collins, and Kei Koizumi. In a letter reminiscent of Roosevelt's 1944 letter to Vannevar Bush that pretty much set science and technology priorities for the next several decades. It created the National Science Foundation and really set our priorities for 50, 60 years. He asked Lander to advise on public health threats, to advise on mitigating the impact of climate change, keeping the country a world leader in innovation, to use science to improve social equity and strengthen the U.S. research enterprise. Additionally, Biden has set forward his $1.9 trillion America Rescue Plan that includes $400 billion for a national COVID-19 vaccination and testing program based on advice from world-class leaders in public health and science in the hopes that science will pull us through this pandemic.
Mm-hmm.
So,
can I also mention that an hour ago, the United States officially got re-entered into the Paris Climate Agreement?
This is fantastic.
That is also really good.
This is the funny thing. So we actually haven't left it yet.
No, we did. We did last year.
No, we
did.
No, we said we were leaving, but there's a year that has to pass before you can actually be.
That happened. We lived through that year, Justin. That was COVID. That was the whole last year.
But my point is that the actual day when we would be leaving is tomorrow. The actual day, the first day we could actually leave it was tomorrow. And the joke was, if Biden wins, it's like it never happened.
Okay, Justin, Blair is now fact-checking your comment.
Okay.
I see her eyebrows in fact-checking mode. So Blair will bring that up. But what has happened today is historic for hopefully bringing science back into a level of import in our government and the policies that are agreed upon by the people in power.
So, Justin, you were close. It was the day after the election, not the day after the inauguration. So we left on November 4th.
For 2 months. Oh, okay. Mm-hmm. So still, it's so we had to rejoin.
It was a short respite, but the important thing is the entire time that was happening, we weren't working towards the goals of the climate agreement.
Well, yeah, but everybody was, because we were all nailing it better than we had based on the.
But the idea is now it is acknowledged again. We have rejoined. We can all work together to meet emission standards again.
Good.
That'd be great.
Still important.
And another important thing to point out was there was a rollback of vehicle emissions, but it didn't roll back California's standard. And since so many states just copycat what California does for their state emissions rules, and the auto manufacturers weren't going to go and make 5 different versions of emissions for California, for United States. So they just made the cars to the California standard anyway, which was the higher standard than the federal to begin with. So nothing happened there either.
Well, I am excited. I am looking forward to seeing what what will be done when we have a fully staffed and fully running Office of Science and Technology policy.
Yeah, this is all very encouraging stuff.
Yes.
So we can get back to where we were, but then the really important part is to then continue moving forward.
Yes. Let's move forward. Keep making
Step
progress.
one, let's get back where we were.
Yeah. All right.
Yeah.
Justin, tell me about these ants that you like.
Oh, how did we say this? Attine ants?
Attine?
Attine ants are farmers. They're farmer ants. They grow fungus as food.
Mm-hmm.
Pretty straightforward, pretty smart, pretty, one of those things, too, when we talk about animal intelligence, here's agriculture at the ant level. So they have some farm hands that help them do this farming. And those farm hands are bacteria. And these bacteria produce metabolites that protect the fungal crop from pathogens. Mostly from other parasites, but those parasites are usually like other fungi that would devour them. Researchers report in ACS Central Science that they have identified the first shared antifungal compound among many of these bacteria across Brazil. And they think it could have some medical applications. So the Attine ants, they originated at some point 50 million years ago in the Amazon as one species, and they have since evolved and spread across South and Central America into 200 different species. All of these ants are doing the fungal farming. One of the interesting things is that the bacteria that they have been thought not to be making the same metabolites, these little helper molecules. So finally, though, they have discovered common ones across the 200 species that the majority of them, at least, from region to region, which point to a conserved purpose. And this is Monica T. Pupo and John Clardy, colleagues, wanted to find out if any of the antifungal bacteria metabolites with that broader distribution have any potential uses. In the study, the bacteria from ant nests at multiple sites in Brazil, the team discovered that nearly 2/3 of the Pseudonocardia strains produced a potent antifungal agent, which they called adenomycin, after the ants. The discovery marked the first time they'd found these sort of broad geographic distribution. What was sort of interesting then, they used it in fighting Candida albicans infection in mice, comparable to zolcaning antifungal treatments that are used clinically. It worked as well, making it actually a potential drug candidate. So here we have this ant that's doing agriculture, and the bacteria it's attracting are creating antifungal agents to protect the fungi that they've been farming that may one day help save human lives. So well done, ants of South America. Thank you very much.
Well done with you and your fun guys. It's like a party. That's awesome. We need all the antifungals and
what a fun place to find it too.
Yeah. Totally. Blair, want to bring us into our next story?
Yeah, I have bad news.
Oh, no. It's okay. We've got hope. We've got antifungal hopes. And now...
Skip your story.
Yeah. Never mind. It's okay.
We got bad news.
No, I have, unfortunately, some real talk from Australia looking at the Great Southern Reef. This is not the normal, just the barrier reefs dying. No. So this is looking at the Southern Reef and specifically looking at the effectiveness of marine protected areas. Marine protected areas are basically like nature preserves, national parks, stuff like that, underwater. They are supposed to be protected space for marine plants and animals so that they can have undisturbed wild space. Because just like with animals on land, if you allow people to do whatever they want in these spaces, the biodiversity will suffer in a bunch of ways. And so this is specifically looking at partially protected areas. So there are marine protected areas, marine reserves, that allow some forms of fishing. So they might restrict fishing on certain species that are of ecological importance or are endangered, or use a specific place as a breeding ground or any sort of thing like that. And so that is their attempt to create more marine protected areas, because it is easier to pitch that and to get it passed and to have compliance if there's some leniency on the rules and it's not just stay the heck out, period. But this research, this is coming from University of New South Wales, found that these partially protected areas basically don't help at all. So fully protected areas, no-take or sanctuary zones have more fish, higher biodiversity of marine life, and they were an attraction to coastal users. So there's also ecological benefits to people being interested in seeing these ecologically rich areas as part of ecotourism, which is a whole nother conversation, which we could get into another time. But what they're saying is that these partially protected areas are considered now by these researchers to be something of a red herring of marine conservation, because they distract from the more effective protection. 69% of marine protected areas are open to some form of fishing. And this research found no social or ecological benefits for partially protected areas relative to completely open areas. The ecological data revealed that fish species richness and biomass were higher in fully protected areas, but not higher at all in the partially protected ones. There were 1.3 times more fish, 2.5 times more fish biomass, and 3.5 times more large fish biomass in fully protected areas compared to open areas, but there was not a difference for the partially protected areas.
So as part of this, like when you have a partially protected area, I'm assuming like you can fish, but the fish have to be over a certain size, perhaps. Otherwise, you have to throw them back, kind of a thing. And then that takes out all the big fish. And so then you start creating a man-made natural selection cycle where all the big fish genes are being pulled out, and you might have fully grown runts that then become the main breeders. I don't know. Could be something.
That could be it. It also probably is just that the marine ecosystem is complicated, and so even if there is a species that is not declining in number, by impacting that species, there could still be a trickle effect on the rest of the food web. Not to mention that if you're allowing fishing activities, period, there is bycatch, there's disruption, there's all sorts of things that happen to the ecological space as a result of fishing.
Plus, you're selecting for something that people want to eat. You're not taking a percentage of everything that exists in this group.
It depends on how the fishing happens, though. I mean, that's always a question. But yeah.
Yeah, but I mean, you're usually going after a human food fish. And so you're not
as opposed to starfish.
All right, yeah. If you were just taking like we're going to take 10% of everything that's there, the food web might still stay relatively intact, and the balances might still be there, and life might still go on as it would. But if you're just depleting 2 or 3 species compared to the rest of it, that becomes a big disruption.
Well, but there's that, but there's also the fact that a lot of fishing techniques are not selective. And that's really the other piece of this, is you might actually be taking 10% of all biomass without realizing it.
I think that would be fine.
That's what I'm saying. I don't think that is the case.
Yeah, I think that would actually have been better than just being selectively taking off the top feeders or the food sources of other things, whatever it is they're going after.
I think it's interesting too, thinking back to your story about pandas from a week or so ago, about the idea of a protected area that's protected for maybe one species as opposed to a broadly protective area that in which there are lots of ecosystems or niches that are encompassed in that. And how does that whole food web, how do all those animals interact with each other? And if you've got your partially protected food, your protected area, you're not really protecting everything in there. And or if you cut it off at a certain point, then things inside get protected and then they swim out and they're not protected. And
Yeah. I mean, I think we had a story not too long ago on the show where I talked about how a marine protected area can actually benefit fish species or marine species that are inhabiting a much larger area than just that marine protected area, because it creates a refuge. And that is, again, it comes back down to just fully protecting specific sanctuary spaces for entire ecosystems.
Yeah. If we're going to do it, let's do it. Let's protect them. Yeah. I don't know. Let's protect a whole ocean.
That would be good.
Yeah. Deep sea miners might have something to say about that, though. But yeah. You want to talk about catnip and cats?
Yeah.
Cats and
Cats on catnip are crazy.
Yes, cats on catnip are totally crazy, and it's really fun to watch your cat freak out when you go ahead and give them a little bit of catnip. And researchers have shown that there is a particular compound in the catnip plant that the cats are responding to, and it activates the cat brain's opioid reward system. This was just published in Science Advances this week. So catnip and silvervine, which is an herb that's even more potent than catnip that's found in Japan and China than in the mountains. So if you want to go ecosystem ecotourisming, hiking up mountains looking for something to really excite your cat, we could look for silvervine in Japan and China. Anyway, nepentolactol. This is the compound that is responsible. Nepentolactol, it gets in there, it activates the opioid reward system, and your cat is actually becoming high. It's a psychoactive state. The researchers wanted to know exactly what was going on there, and there was some other research that found that this compound, this nepentolactol, is also an insect repellent, right up there with DEET in how useful it is. And so the researchers were like, I wonder what will happen if we cover cats in nepentolactol and then stick their faces in a terrarium full of mosquitoes.
Oh, no!
They wanted to know the answer to the question of, do cats just use catnip because it excites them and gives them a high, or do they use it because it also has an insect repellent property? And it turns out that they can't answer this question completely, because you can't ask a cat how they evolved this catnip relationship. But when they had cats, these cats were sedated and they stuck them in the mosquito chamber, if they had been rubbed in nepentolactone, 50% less, half of the mosquitoes, the amount of mosquitoes attacked them. So cats wild cats even, may be using catnip and silvervine, not just because it makes them happy, but because it keeps the biting and scratching insects away.
But is one an unexpected byproduct of the other? That's what.
Right.
So could you distill out catnip that doesn't have mosquito repelling powers and see if cats still mess with it?
Right, if they would still use it. They haven't done that, and that would be a very interesting next step in the research. One aspect of it, though, is they gave the cats naloxone, which is a drug that inhibits the effects of opioids.
This is my next question.
Yes, and the cats are like, Psh, I'm fine. I don't need to rub on the catnip.
Oh, so they ignored it.
They ignored the catnip once they had been given an opioid blocker.
Interesting.
Yeah.
So it kind of sounds like the mosquito repellent is an unintended bonus.
It seems as though it is, and maybe it is this physiological impact, and then perhaps cats.
Perhaps.
Perhaps these cats enjoy the benefits of insect repellents as a side effect, but this is still a question under investigation, but it's very interesting. These researchers, though, have moved forward into patenting a mosquito insect repellent that uses this nepentolactone.
That's going to be great.
Yep.
I'm just still picturing, of course, it's like a Spanish fold cat with the squishy face and the and the like floopy ears, just getting stuck into a box full of mosquitoes face first.
I know. Sloopy, sedated cats.
Yeah,
A box of mosquitoes.
I'm picturing the outdoor barbecue with the tiki torches made from this compound. And so, like, yeah, it's great to keep some mosquitoes away. And then suddenly all the neighborhood cats are coming around.
Yes, and then they knock them down and the whole neighborhood goes up in flames.
Then you need your cat repellent, your coyote urine or whatever that you sprinkle around. It just becomes...
I don't know. Yeah, Gary Lubert is asking, is this getting an Iggy and Ig Nobel? Perhaps. Perchance. Yeah, but it's a fun study into this fun effect that we enjoy with our cats. And who knew there is a little bit of a real benefit besides your cats just getting crazy. Yeah. All right, tell me about these twins.
Oh, yeah, this is a study out of University of Chicago on twins, sets of twins. So it's two twins, because that's how they come. One with food allergy.
A pair.
Well,
Imagine that.
So you're just saying 2, not 4, because at first I thought you meant 2 sets of twins.
So the thing is, there's like
Just
something.
2 individuals
13
related
pairs
to
of
each other.
twins. If it was just a study on 26 twins, you don't know if it's both of them or
see.
just one
Okay.
person who happens to be. It's a very, and there's also 2 of these studies, so it's very confusing.
Oh, okay. Now we're at 52. Okay.
Yeah. So, but what they selected for was sets of twins, 2 peoples, one who had a food allergy, one who did not. So this is results are being published. Were published earlier this week in the Journal of Clinical Investigation. The study grew out of a prior research in the Nagler Laboratory, University of Chicago, on fecal microbiota transplants in infants. So they took the fecal microbes from healthy food healthy and food allergic infants, and they transported them into mice to see what would happen. Turned out the healthy infants, the infants who didn't have the food allergies, were able to pass that on to the mice. Aha! So there's something in the microbiota that is connected to allergies. That was a fascinating study. In this study, this is quoting Catherine Nagler, professor of molecular engineering at University of Chicago. In this study, we looked at a more diverse population across a large range of ages. By studying twin pairs, we had the benefit of examining genetically identical individuals who grew up in the same environment, which allowed us to begin to parse out the influence of genetic and environmental factors from then the microbiota. So after went to a conference, Nagler ran into Carrie Nadeau, and they decided to collaborate on a project. Nadeau had been conducting a study on the epigenetics of food allergies, had already collected fecal samples from twin study participants. Nagler's lab then jumped in, doing sequencing on the samples collected from these 13 pairs of twins with and without food allergies, as well as an additional 5 pairs of twins where both twins had at least one food allergy. Research team looked at which microbes were present in the fecal samples, as well as the metabolites, metabolic products of the microbes. And they also looked at some stuff from the host, the dietary sources. They looked at a whole bunch of, this is Nagler again. We desperately needed biomarkers to understand the immunoregulatory function of intestinal bacteria. Metabolites give us clues as to what bacteria are doing mechanically to regulate the immune response. This approach identified 64 distinct sets of bacterial species and metabolites that are set apart from between healthy and the allergic twin groups.
So we're starting to dig into the
Yeah,
populations
we're
of
drilling down to the population of bacteria and their metabolites that they're creating that are different from one case to the other.
And this gets also at the question of whether it's genes leading to that gut ecosystem, or whether or not it's what you eat.
Yeah.
And so what people have eaten over a long period of time could lead to these discrepancies, healthy gut versus allergic gut, and then we can drill down and take genetics out of the question, right? Is that what's happening?
There was that one study we did several years ago that was all about dishwashers. Do you remember that? Where there were more food allergies in homes that had dishwashers.
Mm-hmm.
This was in I want to say it was a Danish study. It was somewhere Scandinavian for sure. But it was an interesting question of then how much exposure actually has to do with it, which would feed into the microbiome, of course.
Mm-hmm.
Yes, and we was it what's his name? Albert, the physicist at UC Davis. We had him on at the very beginning of the show. And he'd grown up in the Black Forest of Germany, out in the outdoors, and has very few allergies. And I think he was talking about other family members who were indoors but that's been a thing that we've seen over time too. It's indoor kids versus outdoor kids, whether or not you have exposure to animals. So yeah, the more sanitary the environment, the higher the rate can be of having allergies. So what's sort of interesting too in this is then you have, it's nutrition. You are what you eat. That determines everything. That was sort of the mantra for a long time. And the genetics comes along. It's like, no, your genes determine everything. It's not what you eat. That's not enough. It's also your genes that determine how you. And now we're getting to the point where like, whoa, back up. Genes, you got territory. Nutrition, you're also important for some things. But actually, it's the crew on board Spaceship U in your gut. Mm-hmm. It's actually pulling the levers and determining what your health is based on what you eat. So,
well, if you think about how koalas eat eucalyptus, the way that they can get that done is because babies eat their parents' poop. And that gives them the seeded microbiome that they need to eat eucalyptus and process it. So this is, kind of related to exposure. We were talking about exposure before. We were talking about how clean things are in your house. Maybe that's actually, it sounds gross, but trace fecal material that's getting into your body because it has microbiome to seed your gut.
Yeah, you could also just have share the couch with your family.
Sure. That's the easy way.
Anyone who has an animal eats a large amount of their hair. I'm sure I get cat hair on me all the time. I also have my family that I live with, and we are sharing things constantly, and anything that becomes hand to mouth is potentially introducing new bacteria. Even an apple, or, if you don't completely wash a piece of fruit, or, and there's so many ways to populate your gut.
Yeah,
and if you're not somebody if you don't eat yogurts, or if you're not somebody who eats prebiotics, like fiber-rich foods that allow bacteria to grow, maybe you eat a lot of sugar in your diet. These are all things that are going to impact that population.
Yeah, but it's an interesting idea that you could reverse food allergies with perhaps a different microbiota, which is a good time to plug Dr. Justin's Not a Real Doctor Poo Pills.
Yeah.
Allergy free, except for bell peppers. So you get most of the way there. And cinnamon is just, I don't know if it's an allergy or just disgust. I think it's just inherent disgust. with cinnamon.
okay.
That prevents. So this is first author Ryubo of PhD, now research associate professor of medicine at the University of Pittsburgh. To narrow down from thousands of bacteria to specific species as candidates for future therapeutic interventions, one dimension of data is not enough. Bringing together data from multiple dimensions is the key. In our study, we harnessed the benefits of both high-throughput microbiome sequencing and metabolic profiling techniques. We were able to nominate 2 specific species, each involved in distinct metabolite pathways that can be prioritized as potential targets for future research and therapeutic interventions for food allergies.
Yeah.
Now, Verbart says, we can't say that this is cause and effect relationship yet. It looks, but this is one of those.
It could be part of it. It's part of it, maybe.
This is how you know where to look, right?
Yeah.
But we can say there's an
Start.
association with disease and health. So now we can start to ask, what does this mean? Future.
What does it mean?
So future research
Yes.
projects are, of course, being planned on this. But how amazing would it be for all those folks out there who are suffering from 1 or 2 or multiple significant food allergies? Their help is on the way.
Nice. What about people who are suffering from bacterial infections? You talked about your new antifungal from ants, but what about the possibility of either creating new antibiotics or refreshing antibiotics to get over bacterial resistance? New study in Nature Chemical Biology out of the University of Illinois, Chicago. A couple of different labs, Yuri Polikanov and Alexander Menken, have been working together for several years on antibiotic resistance. And they've been specifically focusing on a group of antibiotics that are called macrolides. And macrolides include antibiotics like azithromycin, erythromycin, I think there's some more in there, clarithromycin. They're all the mycins. A lot of mycins. And when the researchers started this recent investigation, they started digging into the structure of the bacterial ribosome. It's been known for a while that macrolides bind to the ribosome somehow. The drug binds to the ribosome and prevents new proteins from being made. So the bacteria can't grow, they can't replicate, reproduce, and ta-da, no new bacteria. Your body is able to defend itself and you get better. But what happens with bacterial resistance? There is a molecular change, a mutation that shifts the structure somehow, so the antibiotic can't get in there and bind to the ribosome anymore. And so they went high resolution and looked really, really closely at what was happening. What they discovered is that the structure of the structure of the ribosome was becoming dimethylated. That means there is a little part on one of the amino acids that's in the ribosome that got 2 methyl groups, 2 CH3 groups added to it, where before there was none. And as they were looking into this change and what that meant, they discovered that water is the binding agent for the antibiotic to be able to bind to the ribosome. That when those methyl groups aren't there, water can come in and, because of its polar nature, bind to the ribosome, and then the antibiotic grabs on. And that's how the catch is made. But the mutation that allows that methylation then keeps water from being able to bind, and the ribosome can no longer attach. And so now these researchers are thinking, it's a very exciting thought, they are thinking that they will be able to move forward and hopefully be able to create new antibiotics, or be able to go in and refresh antibiotics to enable them to get around this methylation problem so that water can remain the binding agent. But it's so great. It's just amazing to me. This one little change. It's not a huge change. It's this one little change that just keeps water from getting in there. It's like, no water. Nope, not today, water. It's amazing. And that is what is threatening an entire group of antibiotics from being useful anymore. But now we have a direction to go. And this is a completely different model from what has been hypothesized in the past. So it's like this completely new direction to go. But new antibiotics could be great.
Nice.
Yeah. Dig in there to those molecules. Dig into that science. This is This Week in Science. We are here with our science. Thank you for joining us once again. If you are interested in a TWIS shirt or a sweatshirt, I've got a sweatshirt on today, you can head over to twis.org and click on the Zazzle store link. Browse our store and help support the show. Thank you for listening to TWIS. All right, everybody, you ready to come on back and talk about COVID?
Yay! Do you have good news for us this week?
Nope.
No. Oh, well.
Nope.
All right. Proceed.
Yeah, thanks. Okay. I would love to give you good news, but the news just keeps kind of being a little depressing. A variant has been discovered in Brazil and was reported this last week that this variant, we've talked before about some of the variants that were discovered in the UK and South Africa. This new variant, though, instead of making transmission higher, what it seems to have done is escaped from antibody immunity. What this means is that people who have had the virus, who have had the disease previously, were getting infected again because it had escaped from the antibodies that their bodies had produced in response to the infection. And this is one of the issues that we have discussed with relation to this virus, is that as the vaccine effort is going out, we're trying to race the clock, the mutational clock, to get people vaccinated so that the virus will go down to low levels in the population and not really be as much of a problem. As we said, the more that it spreads, the more that it mutates, the more the chance that it will escape from vaccine immunity, which is a very similar thing. But the vaccine immunity is very specific to the spike protein.
Well, and I mean, if we had done a better job of controlling community spread, then even if there were localized variants, then you could kind of isolate and control spread of that specific variant. But because it's just kind of all going everywhere unbridled right now, it's very difficult to isolate and restrain spread of new variants.
Yep, it is very difficult. And so I'm hoping that around the world, vaccine rollout can go more smoothly, more quickly. And also here in the United States, that we can institute more tracking and testing so that we are more aware of what kinds of variants are showing up in our population at a much more high resolution level than we currently are able to see. It would be pretty amazing. But regardless of all this, human behavior is the cause of the spread of the virus. People to people moving around, that is how the virus spreads, right? We go out, we breathe on each other, and virus. So there have been some studies, one coming, let's see, this study here, one study out of the Journal of Econometrics looked at the causal impacts of masks, policies, behavior on early COVID-19 pandemic in the United States. Their conclusion was that if the United States had adopted a national mask mandate early in the pandemic, 47,000 lives could have been saved before May 1st. And if you want to extrapolate that out, do what we're dealing with right now, you can do that math yourself, but I don't feel the need to go into that. And then additionally, a paper in the Lancet out this week that's separate from a mask mandate, but just relied on personal reports of people's mask use and social distancing. They were able to correlate those behaviors down to local regional levels and improved control of the SARS-CoV-2 viral spread in those areas. So the more people in a particular region reported wearing masks and social distancing, the better that region seemed to fare in terms of the number of people infected. So I don't know how to say it any other way. As long as the vaccine rollout is happening, it is not the silver bullet. There was another study that I couldn't find this, that I lost the link to it, but the logic makes sense. You can save more lives by people continuing to wear masks and social distance than by focusing on high priority vaccinations. When you vaccinate somebody, you are vaccinating and protecting that one person. Mm-hmm. When you wear a mask, you are protecting everyone you come in contact with. So the math there, I just want to put that in your mind when you're going out there, and maybe you can, as the vaccine rollout happens, talk to your friends, talk to your family, try and find out, if they are hesitant about masks or social distancing, what could help them to potentially adopt that behavior a little bit more, because we can help each other.
I mean, I get it. Everybody's sick of it. I'm sick of it. It sucks. It all sucks. We all wish we could be with each other. We wish we could eat out at restaurants with our friends and family. I get it. But you can't just give up. You can't just give up. And I am seeing that. And it's really frustrating. And I won't go on and on about it. But in my community, in my apartment complex, I think we had like a 95% compliance of social distancing and mask wearing for the last 6-8 months, ever since they started telling us to wear masks. But in the last month, it has dropped off where it is unusual now if I see someone wearing a mask and keeping their space from me when we are walking around.
Right now is like a really bad time.
The worst it's been.
It's the worst it's been. It's the worst it's been in numbers, but it's also the worst it's been in compliance that I have seen anecdotally. And it really just feels like people are just sick of it. They're just giving up. But you can't give up, because like if someone hasn't died yet who you know, that doesn't mean it's not going to happen. That just means it hasn't happened yet. And you need to protect those people around you.
Yeah, protect the people around you.
It's very frustrating.
It is frustrating, and it's kind of like I've talked about it as an analogy to like running a marathon before, but maybe it's like rolling a stone up a hill, and we're about, halfway, 3/4 up, and past the halfway mark, but we've been pushing that stone up the hill, and suddenly we're just like, I'm tired. That stone is just gonna roll right back down to the bottom, and we're gonna start all over again. And I don't want to do that.
And my rant remains the same as it has for the last 9 months, 10 months. Don't push the stone. What are you doing outside pushing stones up hills? Stay
home.
6 weeks.
Yeah.
Everybody, and it's done.
Mm-hmm.
Yeah, if we could.
Stop.
If we could.
It could be over. Oh, we can't afford it. How's that working out? How's that working out? The half measures. How's that working out for everybody?
Well, I mean, people wearing masks. Here's Stephen Rain saying, I kind of like the mask. Haven't been sick for about 1.5 years. And we are seeing people wearing masks and social distancing. Flu rates are going down. Common cold rates are. This is working a bit. It's just this virus is more infectious than those things. And so that's the problem. But we just have to keep it up.
Mm-hmm.
You can do it. We can do it. We can do it by helping each other, having conversations that are hard conversations. But we've come this far, or you've come this far. Your family, your friends have come this far. Don't let them stop now.
Yeah, don't let everybody. I mean, just picture being like fatigued and then like, I'm so tired. I'm gonna go commit suicide. That's what I'll do today. It's just a terrible idea. Don't give up on being alive. Hey,
Don't
wait.
give up on it.
Now, I didn't get to comment on this yet, but the first story you brought
Mm-hmm.
seemed to indicate that even if you've been vaccinated, you might at some point need to get a new vaccination of a different vaccination later because
Possibly.
this is
It depends.
going
Yeah.
to keep going.
Possibly. It's not a for sure thing, but these mutations happen all over different parts of the virus. And some of these mutations in the B117 virus have led to increased transmission. Also, the South African virus has mutations in what's called the 501Y.V2 variant, and its mutations at E484K and K417N. These are just locations in the virus. These are changes in the surface protein spike and have been shown to reduce how well monoclonal antibodies combat the virus. Additionally, there is also E484K, which reduces the potency of convalescent sera from some donors tenfold. So these different variants, these different mutations, they have differing effects. It depends if we are using the spike protein for the virus, for the vaccine at this point in time. That's what all of the vaccines are the spike protein. So if mutations occur in the spike protein that change it enough that your body no longer recognizes it as what it has created protection for against, then it could potentially escape. So far, and the thing about like natural immunity is that your body could create. It's like I'm going to create an antibody for the finger. I'm going to create an antibody for hair. I'm going to create an antibody against this thing. Your body's just creating whatever it sees and comes in contact with. It could create an antibody for.
But in the lab, it's been escaping.
It can,
Okay.
but at this point in time, the vaccines that we are rolling out are still good.
For
We are
most
good.
of the virus. Yeah. Still go get the vaccine. The virus in your neighborhoods.
Go get the vaccine. Yes.
Vaccine.
Yes.
It'll work just fine.
It will work. It still works. We may have to change some of these vaccines to reflect mutations in the future. This could be a yearly vaccination, depending on how fast mutation rates go. Who knows? We don't know enough about this virus yet.
Which, reminder, that's how flu shots work. So that's already
could be
something
getting
we
our-
do.
Yeah, we keep getting our COVID and flu shots all in one. Maybe. Who knows? Who knows? But let's move on. Let's move on away from our sad, icky COVID news. This is This Week in Science. Thank you for spending your time with us. We do appreciate you being here for these discussions and for science. If you like TWIS, please share TWIS with a friend today. As we come back now, welcome back now, you hear? It is time. It's time for that part of the show that we love to call Blair's Animal Corner.
With Blair.
Dunk.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
Yeah, Blair.
Speaking of icky, I have a really icky story, but it's actually really cool. It's about beetles. Beetles, beetles that are scavengers. So just like opossums, maggots, vultures, they are competing with all these other animals to eat dead stuff. And this is a recent study from University of Connecticut looking at burying beetles, which are pretty big beetles. They're about an inch long. They're black with orange markings. And they have a really cool way of packaging dead stuff for their babies to eat later. They actually spread gut secretions all over a carcass, and they roll it up into a big, delicious flesh ball covered in goop.
That sounds super appetizing, Blair. Thanks.
They'll take a dead mouse, they'll take a bird, they'll dig a hole, they'll bury it, they'll pluck its fur or its feathers, they'll roll it into a flesh ball and cover it with delicious goop.
Blep.
But why do they do it? So it has previously been assumed that it has to do with delaying the decomposition of this food. So they're basically keeping it fresh, keeping that freshly dead stuff nice and fresh. And so the new kind of hypothesis was that actually it was to reduce competition amongst scavengers as well. So the way that they kind of tested this was so anecdotally they kind of thought that there were less maggots, there were less other creatures kind of going after this dead decomposing goopy meat. Yum. But so the way that they actually tested this, scientifically, is they collected the gases wafting off the dead hairless mice. And the researchers then compared the gases from those that were preserved by these beetles to those from untouched carcasses. The beetle prepped carcasses gave off much less of a gas that they describe as onion smelling than and that usually attracts the bearing beetles to the fresh remains. They also discovered that an increase in another gas from decay that is known to deter insects that feed on dead animals were building up on these prepped carcasses. The next step, of course, was to drop off dead mice in a Connecticut forest and to see how they were found, if they were devoured by other animals in the forest. And they found that the beetles' rivals were less likely to discover the ones covered in goop.
Goo.
Yep. So here's why this is interesting. Kind of like the catnip story from earlier. Is this a dual purpose thing that these beetles are doing? Is one a side effect of the other? What is happening here? Did they spread it with this goop to preserve it, and then it just so happens that it became less desirable to other animals? Or were they hiding it from other animals and it also, surprise, preserved the meat? Or is it a dual purpose thing from the start? We don't know.
That's always one of those questions. Yeah. Did it all happened together, or is it, yeah. They're like,
And
oh, the goop protects it. Great.
Dare I ask, should I cover my food in goop?
To protect it from Brian? Yes.
No, to preserve it is more what I was thinking.
Yes.
Yes. I'm sure if I
is
covered it in smelly goop, then nobody else in my home would know it existed, including the dog. But really, it'd be about preserving the food, you see. So actually, no jokes aside, you could potentially see a practical use of this information, because you could, in theory, find something about this goop that is so good at preserving meat that you could apply to human food.
Right. Beef jerky without being jerky-ish.
You could also find something in the smell that deters other animals and use it in some sort of pest abatement.
Is it
farms
or is
or
it
other
just
things.
masking the scent of the decay from the thing it would want to eat? Right?
So
Because
that's a good question. Is it masking it? Is it converting it? Or is it eliminating it?
Right.
Sounds like there's less of one smell, but there's more of another. So it could be both.
But if you've got something big and you need to eat it, so we know that some animals like mountain lions will bury their dead. They will cover dead animals in a bit. They'll eat a little bit, cover them up, and then come back and feed off of the corpse for a while. So, if it's sitting there and it's being preserved and it's also preventing it from being eaten by other animals, you're storing food. It's like a refrigerator or a cellar.
Yep.
You're putting it underground. Yeah.
Yeah.
This is good. But the goop, I want to know what the goop is made of.
So do I.
I want to know what is in the goop. Because in
the
cooking, have you ever done the where you put you take duck fat or chicken? You can take chicken fat or turkey fat, but you take duck fat and you overnight you sit, or maybe like a couple of days, you sit like a chicken or something in it and let it sit for like a day or two, and then you cook it at a very low temperature, like super low temperature, and it's the chicken is amazing. It's delicious. I guess
I'll
you
take
don't
your
know
word
what
for it. No.
Yeah, Miss Vegetarian.
Yeah. Yeah. There's that, but also just that sounds like salmonella soup. Anyway, it's fine.
It's not. No, the fat preserves the meat for a while, and so it sits and preserves it a bit, but then you cook it, and there's something to the goop that you put your bird in and cook it. It's confit.
Inuits,
that's what it's called, confit.
The Inuits in Greenland have a fermented bird dish, which I think consists of digging a small hole, throwing the bird in it, putting a rock over it, and coming back a month later or something like that.
Oh my God.
Yeah.
Oh boy.
There's like lots of ways to prepare the things that you eat. We humans are no different. I'm sure actually that beetle goop food is probably more sanitary than a lot of things that humans do.
Probably. I want to know what's in the beetle goop.
Well,
Hopefully
we'll find out
Exactly.
someday. And the chilling conclusion. Do you want to be further horrified? I have another horrifying story for you. Okay, this is about electric eels hunting in groups.
That's not scary.
Pack hunting electric eels.
Yes,
Yep.
so this is a study from Smithsonian's National Museum of Natural History. And this is looking in the Brazilian Amazon River Basin. And they discovered a small river-fed lake filled with more than 100 adult electric eels, many of which were upwards of 4 feet long. Of course, electric eels are not true eels. They are a type of knife fish.
Wow.
And they have previously thought to be solitary creatures. They witnessed the eels working together to herd small fish, tetras, into tightly packed balls, and then groups of 10 eels would split off, 4 to 10 eels would split off, form cooperative hunting parties that looked kind of like wolf pack hunting or killer whale pod hunting, and they surrounded the prey ball, the ball of tetras, and launched simultaneous electric attacks, which would stun the tetras into submission. Actually, it would stun them so hard that they would fly out of the water and smack back into the water, totally stunned, ready to be eaten. And so this is,
this is a stunning finding.
It's stunning indeed.
Yes.
Shocking, even.
Shocking.
But the crazy thing here is, first of all, pack hunting in fish, very rare. There's only been 9 species of fish seen to do this on planet Earth, possibly because we're not very good at studying fish in their natural habitats in a lot of ways. There's a lot under the ocean we don't know about. So perhaps just we haven't found them yet. But ultimately, this is a new thing. There's only 9 other species at this time that have been found to do pack hunting. And they first saw it, they thought it was totally anecdotal and maybe just a fluke in August of 2012. But then as they were studying the Amazon River Basin, in October 2014, they found it again. They settled down, they logged 72 hours of observations of these eels in this river. And this is at dawn or dusk in the twilight hours. They would interact with each other. They would begin swimming in a large circle. This churning circle would move these tetras into tighter and tighter shoals, into these balls. They would herd them from deeper ends of the lake into shallower ends of the lake, and then some of them would break off and do this shocking thing, and the tetras would go flying and all this kind of stuff. It took about an hour and contained between 5 to 7 high voltage attacks. So this is something amazing. That, yeah, now it's like 2 points make a line, right? So like at this point, they feel like it's definitely happening. How widespread this phenomenon is kind of up to question, because in terms of interviews with locals all over the Amazon River Basin, there hasn't really been reports of this. And you would think that 100 eels shocking in groups in kind of shallow water, like 3 feet deep water, you'd hear about that from locals. So and there's a chance, yeah, they really hadn't. So there's a chance that this is pretty localized, pretty individual, pretty unique, but it also is possible that people just aren't seeing it. So this is a whole new thing that they're looking at. They've launched a new citizen science program to try to locate if there's more of these aggregations happening. And this citizen science project allows users to report sightings and log observations themselves in Brazil. And their next step is they hope to collect tissue samples and mark individual eels with radio tags to understand if they're related at all, if there's a genetic information passing thing happening, if these groups are related to each other and that's where they hang out, if there's hierarchy. And they want to take direct measurements of electrical discharges in order to see their maximum voltage, to determine whether eels might also be using low voltage shocks to communicate with each other, to orchestrate these kind of complicated events.
Orchestrated. I'm looking at that little bit of video that we placed up there, and you can see there's the fish sort of in the middle getting circled by one group of eels that kind of has them, it looks like cornered up against the shore somewhat. And then on the outlet, there's a couple of eels hanging back. And why are they hanging back? It looks like in case anybody breaks the perimeter, if the perimeter starts to break, if they lose containment, they have some backups that are waiting, sort of like a defensive backs or a safety in a football game, like waiting back there for if the run game escapes the front line, they're ready there to regain containment. And it really looks orchestrated. Like, wow.
Eel-castrated.
Yeah.
Real strategy looks like it's being
indeed. Yeah. The last step of the study also is they do plan to collect 8 to 10 eels and bring them into a facility in Germany where they can conduct controlled tests and that then they could later replicate in the field as well. So this seems like a pretty well-designed effort to figure out exactly what is going on here. And one of the researchers does mention that they'd been shocked by these eels plenty of times in their scientific processes. And the shock lasts about 0.02 of a second, but is enough to cause a painful muscle spasm that could knock a person off their feet. And this is one eel. So if you have
imagine a whole bunch.
Yeah, it'd be pretty crazy.
Down.
And if you've ever been to a fish hatchery, when they are collecting fish, they go in with electric paddles. The people who run the facility, they have these massive electric paddles that they push in, they put into the water and go zzz, and zap the water, and you see the fish jump out of the water, and then their belly up on the surface, knocked out for a moment so that they can be collected for eggs and other things.
Yeah. Well, so let's hope that the Smithsonian National Museum of Natural History Fish Research Associate, C. David de Santana, can use his expertise, his previous shocking experience, and his now laboratory electric eels to find out exactly what's going on.
Take it to the lab.
Do people eat the electric eels? Are these being hunted otherwise? Or do people avoid them?
I don't know. That's a good question. I would
leave them alone.
I'd leave them alone. But the thing I was thinking about, like maybe why nobody's like witnessed this behavior is if they are a food thing and you're coming across and it's like, oh gosh, there's like 7 of them right there. I better start pulling them out of the water with sticks. You disrupt the behavior pretty quickly.
Right.
So, I mean, a very quick Google, I have not fully vetted this, makes it look like knife fish and electric eels specifically are not often eaten because there's not a lot of suitable flesh to eat.
Okay.
So, anyway.
Because of all those electric wires inside of them.
Yes.
The battery.
Yeah, you
wires.
put her on the battery for sure, the transistor. Sometimes the battery leaks because they're like in salt water, right?
Yeah. No,
anyway.
It's no good.
I have one last story in the animal corner that I had to add last minute because this was breaking news. And this is that the first dinosaur butthole ever discovered.
Well, you can't say butthole.
Has been analyzed. I'm sorry, the cloacal vent of dinosaurs.
What's a cloacal vent?
It's a butthole.
It's like a bird butt.
Yeah,
hole.
so a cloaca is the all-in-one, as it is. Mm-hmm. So it's
the common urogenital tract.
Yes, so it has the reproductive stuff, it has all of the excretory exits there, and it actually comes from the Latin word for sewer. Mm-hmm. So that's the cloaca. Reptiles have it, birds have it, amphibians have it, fish have it. Lots of animals have cloacas. We're one of the unlucky ones that do not. The cloaca is
it's not unlucky though.
Is the muscular chamber. Usually what you see actually superficially is called a vent, which is where there's the parting in the scales or where there's the area that opens to allow for the exit of various items. So anyway, that also means that there's not a lot of apparent physiological difference externally between males and females. There's no external genitalia. So it's a lot harder to tell, for example, with dinosaurs, unless there's some other sort of dimorphism, like males have these giant horns and females do not, or something like that. It's a lot harder to tell male and female dinosaurs apart in paleontology because of that. So anyway, regardless, this is just the most perfect preserved cloaca. It's the best these researchers have ever seen. This is not a peer-reviewed article. It was not published in a journal. This is really just the breaking news that this thing has been discovered and looked at. So what I'm about to tell you are ideas from some scientists based on observing this perfectly preserved cloaca.
All right.
So first of all, there are these two small bulges by the back area, which are very reminiscent of musk or scent glands that we currently see in crocodilians. So that's a good idea that potentially these dinosaurs had that. So the type of dinosaur, which I did not say, is a Psittacosaurus, which is a bristly-tailed, Labrador-sized, which already I'm in, horn-faced dinosaur, and it was a relative of Triceratops. They lived in the Cretaceous period from about 145 million years ago to 65 million years ago, somewhere in there, and it was found in China, this specimen. They were previously studying it to look at skin color. They were trying to find proteins and other identifiers where they could extrapolate what color dinosaurs are, because contrary to the bed sheets, books, and sweatshirts I had as a child, and my Chuck Taylors, they are not orange and purple and pink, although I wish they were. No. Anyway, this cloaca on this little Labrador-sized horn-faced dinosaur, the Psittacosaurus, was so well preserved. So first of all, they think they saw these scent glands, but also they were able to see there's actually a preserved poop in it.
A coprolite.
Yes. So they think they might be able to get some information out of that.
I'm sure they will.
And the other crazy thing is that the outer regions of this cloaca had a darker shade of melanin than the rest of the underside of this dinosaur. So either it was a type of visual display, like in baboons, or potentially, it could provide antimicrobial protection. And this is where I was not aware of this piece of science that exists, which is that in humans, there's melanin in certain parts of the body that never sees the light of day. I know where your brain is thinking. That's where my brain was thinking as well. But actually, they are referring to the liver. The liver is full of melanin, and that somehow helps it with microbial infection. So that's the other idea here, is that
outside.
Yeah. So that's all very interesting. But anyway, just this perfectly preserved cloaca. They don't know if it was a male or a female because the soft reproductive tissue that would be inside were not preserved, and so they're not sure what sex it was. But they
got
do
the
think
vent, and they've got this melanin, and
they have the scent glands.
Yeah.
And the other thing is based on the shape, location, all this other stuff. They think that they also had copulatory sex. So
not just egg laying or
not just a cloacal kiss.
A cloacal
so
kiss.
You have the cloacal kiss,
Yes.
which is what birds do, which is they just press their cloacas together. But then there is actually copulatory sex, which is when an organ exits the cloaca completely, and then just more standard stuff. So anyway, that's what they think, but who knows? And again, this is all pre-publication, pre-peer review. This is all just kind of conjecture based on this perfectly preserved butthole.
Well, you can't call it that because it's like the Swiss Army knife of all the down there stuff.
But just that there was a poop in it, I think I'm allowed to call it that.
There was.
I guess in this case, that's the function it was probably about to render next.
Come on, you don't want to be on record here as being anti-fun, all right? Because this was fun.
When it just in the science fun police.
I know, what happened?
I love the idea also of how many birds have displays where they puff up their tail feathers, or they have some kind of display dance where their tail feathers go up, or they expand their wings, where there's a dance where they strut their stuff and show off those tail feathers. So
yes.
Yeah, I'm imagining this Labrador-sized, bone-faced dinosaur now doing a dance display and showing off its hindquarters to a potential mate.
Yeah. I love it.
Could be. I don't know. That's a fascinating. More dino butts. We need more data on this. Yes.
Yes. I want to hear more about the poop. Am I the only one who wants to hear more about the poop?
I want to hear more about dinosaur sex. But that's just my take on. Yeah, so did they analyze the poop?
Not yet.
Oh, that's going to be awesome.
I can't wait.
The poop will get analyzed, and it will probably have metabolites of hormones. It'll probably have evidence of food that was eaten. There'll be so much information in that poop.
Yep.
That's good. We will know what that dinosaur had been up to. Yes.
Can't wait.
I don't know. And maybe if they have other bones, they could, I don't know what the rest of the fossil is like and what they have, but if they have other individuals, there's the possibility of determining whether or not it's male or female based on comparative anatomy and and pelvic shape and things like that. So there, even though the soft tissues of the genitalia have gone away, there is potentially other information out there.
And I know they have to use the fossil for real science, but when they're done, may I just suggest candles or chocolates? because those are things that currently are done with the derriere of individuals. Do you not know about this?
What are you talking
This
about?
is a thing that
What?
No, it isn't.
yes, it is. We'll talk more about it in the after show, but there's impressions that are taken from certain areas
Yes.
to make chocolates
The
or
chocolates.
candles. There was a lot of
Yes.
news this
Okay.
week about
What are you
candles
saying?
made out of
Valentine's Day
particular
is coming up. That's
celebrities'
why. Yeah.
nethers also. So
Oh my God.
I'm just saying, how cool would it be to have a
Nope.
dinosaur cloaca candle? I would buy it. That's all I'm saying.
Justin is rubbing his eyes.
I don't
I just
understand. I need more coffee. I'm not following anything. What are you talking about?
Don't
It's
Don't go
an
anywhere.
taxidermied candle holder that you're
No.
talking about?
no.
I'm very confused right now.
Justin, I'll send you some links.
We'll talk more in the after show. Justin, don't go anywhere. You're coming up next. Right now, I have to say thank you for being a part of This Week in Science. Thank you for joining us for this show. We hope that you are enjoying it as much as we are. And if you are able, head on over to twis.org and click on the Patreon link to become a supporter of TWIS. No. TWIS.
Twish. Yeah, you know, Twish.
TWIS. TWIS. Yes. To be a supporter of TWIS. Your support helps us do what we do every week, and it also helps to bring a somewhat sane perspective to this crazy world of misinformation. Hopefully moving forward, a little bit less fake news. I don't know, but with your help, we can reach more people and grow the show and continue to do what we do, bringing you the show every week. And at $10 or more a month, we will thank you by name at the end of the show. So head on over to Patreon, click that link, and select your level of support today. Thank you for all of your support. We can't do what we do without you. All right, Justin, it is time for you to tell us about brains. Did you want to talk about brains?
Yeah, I think there's a sort of interesting story. This is going to talk about the human brain specifically, which has about as many neurons as glial cells. The glial cells are then broken up into four major groups. You have the microglia, astrocytes, NG2, and oligodendrocytes. How do you say it?
Oligodendrocytes.
Oligodendrocytes.
Oligodendrocytes, yes.
So what do oligodendrocytes do?
They are support cells for the brain. These are all astrocytes. Oligodendrocytes are part of the brain's immune system. They are part of short distance electrical transmission. We are learning so much more about these cells that we just thought were like packing peanuts in the brain for a really long time.
Right, and so the oligodendrocytes, that's the insulation tape around the axons, around the electrical current sending ones. The wraparound axons, which are the extension nerve cells which send the electrical impulses. This wrapping prevents short circuits, accelerates signal forwarding, and thus we have very few forest fires started by axons because they remain well inside.
Yeah, so these are the when they wrap the neurons, they are called myelin. It's the myelin sheath.
Yeah.
And that's the part also of the neurons that gets damaged in multiple sclerosis.
Yes. Yeah. This is Professor Christian Steinhauser from the Institute of Cellular Neurosciences, the University of Bonn, Germany. We have now been able to show that oligodendrocytes play an important role in the distribution of energy-rich molecules, compounds, nutrition, that they're actually doing more than just, even though they're not sending electrical currents themselves, it seems like they are transporting partially, the converted sugar, the energy-rich molecules that power these neurons, that keep them alive. This is the quotey voice, Christian Steinhauser. This is apparently especially true in a particular brain region, the thalamus.
Ooh.
Thalamus, aka the sort of like the router of the brain. It's like all your sensory signals come in from the eyes, the ears, the skin, and then it gets forwarded like an email that you program that you haven't used forever, but you still want to get some of the emails from it, so you have it forward to your new one. Forwards everything to the respective responsible centers in the cerebral cortex to do the actual analysis of the sensory input. That's when we realize, information is coming in when it gets to the final destination. But so the thalamus is controlling sort of like this routery sort of thing. So it's long been known that astrocytes can form close connections. They build intercellular networks through tunnel-like coupling. Molecules can migrate from one cell to another through these gap junctions. A few years ago, Steinhauser and colleagues were able to show that there are also oligodendrocytes in these networks in the thalamus. About as many as astrocytes, the cells form a huge network in this way, which neuroscientists call the panglial network. That means across the glial or comprehensive glial.
Mm-hmm.
In other regions, however, networks consist predominantly of coupled astrocytes. We wanted to know why this is different, says Dr. Camille Philippot, whose quotey voice sounds very much like Steinhauser's. Our research demonstrates that the high energy compounds travel through this network from the blood vessels to the synapses, and the oligodendrocytes seem to be indispensable in this process. So they tried it in mice, and these mice energy molecules were no longer able to reach the synapses in sufficient quantities. Same was true if the astrocytes lacked the appropriate connection links. The thalamus apparently requires both cells for transport, Steinhauser has concluded. So it's just an additional function to a part of the brain. The little wrapping, it's doing more heavy lifting than we thought it was. It's more than just insulation.
I feel like with the brain, that's the more we study, the more we learn, the more it's like, it's so much more complicated. There's these cells are doing this thing here, and they're doing this thing here, and yeah, it's
the more we learn about it, it scares me. It feels more like a bunch of dominoes. Just like you mess one thing up, and it all kind of.
There is a bit of that, but because of redundancy and the networked nature of the neurons, there's wiggle room.
Yes. There's fail safes. There's other stuff. It can compensate given enough time, but it does feel very interconnected and intense.
But findings like this, the glial cells that we used to think really didn't have much of a function at all, to discover that they have these really important support roles that allow for normal neural transmission to take place, that they are there to support and make sure that all the that you need nutrition, I've got nutrition. You need a sports bar, I got that for you. You know, these are like the glial cells are there like the cheerleaders at the side of the road at the marathon. Here's your energy juice. Okay. And they're really able to do more than we thought once upon a time, and they are involved in a lot of disease. And by understanding these aspects in their support roles, we'll be able to understand better where things go wrong and how we can fix them. Brains.
Next story, lasers. Can it get any better than lasers? Yes.
Depends. Yeah, where are they getting pointed at?
Better lasers. Pulsed lasers. So pulse lasers is exactly what you think it is. It's a laser that's going on and off and on and off and on and off, emitting light for these very short periods of time, blinking on and off. They actually can have this advantage of being able to focus more energy than a continuous wave laser, whose intensity is kept unchanged. I don't really understand why that is, but you can go YouTube it on physics, learn about lasers if you want. Digital signals can be loaded into a pulse laser. Each pulse can be encoded for a bit of data, and you can transmit data with this. The higher the repetition rate, of course, the more data quicker you can transfer across the thing. So far, though, optical fiber-based pulsed lasers typically have had limitations in increasing the number of pulses per second above the megahertz level. So it's cool, but it's not really going anywhere. It hasn't for a while until Korea Institute of Science and Technology, KIST, which is kind of a great acronym for anything, announced that the research team led by senior researcher Dr. Yongwon Song at the Center for Optoelectronic Materials and Devices was able to generate laser pulses at a rate 10,000 times higher than the current state of the art.
Whoa.
Yeah.
What did they do?
Oh, well, this achievement was accomplished by inserting a additional resonator containing graphene into a fiber optic pulse laser oscillator that operates in the domain of femtoseconds. They put a little thing in the middle that helped filter the signal. It sounds like almost like a condenser. Research transmission and processing speeds are expected to increase significantly by applying this method to data communications in the future. So it has to do with something called a Fourier transform. Mm-hmm. If a resonator is inserted into the laser oscillator, the wavelength of the pulse laser is periodically filtered, and thereby modifying the pattern of laser intensity change. Why does that work? Get a degree in physics and tell me, because I don't really know. But here's that. Well, you just need to know that it went from a megahertz to 57.8 gigahertz repetition rate, completely overcoming limitations previously on pulse lasers. It's also the additional characteristic of the graphene they're using, such that heat is locally generated when the laser is absorbed, was exploited then to turn the characteristics of the graphene resonator by applying an additional laser to that device. So lots of lasers, ultra-fast, pulsed laser resonating technology. So point is, things can be faster, smaller, quicker, meaner. It's going to increase the Googling speed. At this point,
we don't need 5G. We need 10,000 G.
It's going to increase how fast you can confuse your cat with a laser pointer. No.
There are still things that exist that take long rendering or downloading times. There still are these places that you can get into for data transfer and the rest that take what people today think is a long time. But by oldie people, it took a minute. And then, the kids today are like, a minute of my life is gone where I'm not playing, a video game on my phone. Yeah, but 10,000 times faster does sound like an incredible shift. Especially if it can be, if we're getting into the age of sort of machine learning, and we're talking about data sets that would require that even being able to feed the projections that we have for what machine learning is capable of, just being able to feed that beast the data that makes it worth having machine learning is difficult. It's impossible at this point.
Yeah, and then if we also think about from a science data transfer perspective, we have limitations related to how much information we can take from our large data installations like LIGO and Virgo, like CERN, like so many other of these massive scientific experiments that are producing huge amounts of data. And if you can transfer them more quickly, that would make cloud computing for a lot of massive data computational efforts potentially be easier. I mean, I'm just imagining how that could work. Additionally, we know that not quantum computing, but thinking about doing quantum communications and taking data from and creating quantum communication networks, perhaps this could impact that as well.
Yeah.
Make it more. That's cool.
Yeah, it's also something that I've seen in all the fields working in biometrics around genetic data. There's a tremendous amount of information that is being worked with as the raw material.
And if we ever have to download the entire climate change database from the government ever again, it'll happen faster.
Yeah.
Gosh, I hope we don't have to do that.
I sure hope so too. Hope
I
not.
don't think it'll be a problem for a while.
Yeah. Speaking of data and what we do with it, how about teaching computers how to deal with their own data? How do we do that? Well, researchers have been advancing a particular paradigm of AI algorithm training that is known as, wait, let me make sure I've got my words right. That is, there we go, generalized adversarial networks. That's what it is. And classrooms of the future might have personalized curricula for students created by algorithms that learn themselves from these generalized adversarial networks that train neural networks to solve problems and do things. In a generalized adversarial network, what happens? You get adversaries. You set up one neural network to be the trainer who's going to give something, an image or a problem that's going to start the training process. And then you have the learner who is going to try and learn. So, and it's adversarial because the training algorithm gets harder as it goes or tries to challenge the learning algorithm to actually learn and do better. It's kind of like education as it works in humans. And that's really interesting because this is with neural networks anyway. So these adversarial networks, you have the trainer and you have the learner. These researchers just published or presented on their new training paradigm. They're from University of California, Berkeley, that's called paired. And they couple their AI with an identical AI that has a different set of strengths. So you have the antagonist and the learner. And then they have a third artificial intelligence algorithm design a world that's going to be easy for the antagonist, but hard for the protagonist. And so the task uses these two agents back and forth to challenge a little bit. And it gets personalized based on, it's not just here, do this task and not knowing what's going on. The training antagonist actually, oh, you're not getting that right. Let's fix this process a little bit for you, and we'll help you learn by figuring out what exactly it is you don't understand. If you could imagine the AI talking to each other in this particular way. So they had their programs reach a destination by navigating a 2D grid that had a bunch of solid blocks in it. And the agent, the AI program has to get better at being able to go through that maze, basically, through trial and error. But when the maps were too hard to get through and the AI couldn't solve it, they used this paired procedure. And the protagonist attempted some difficult mazes. If it trained using the two older methods, the protagonist didn't learn anything. But with paired, it started solving the mazes. So it started actually learning when it wasn't learning before at all. The researchers used a version of paired to teach an AI to fill out web forms and book a flight. And with traditional neural network adversarial training, it failed all the time. But when used with the paired method, it succeeded about 50% of the time. So this paired technique, they ended up setting it to an AI on 225 problems that no computer had ever been able to solve, and their agent solved 80% of those problems.
Wow.
That had never been solved before by computers. And these are not, these aren't problems that are like, these are weird computer problems that computer programmers try to use to determine whether or not their program is working. But the fact that they were able to solve these problems that had never been solved before using this new training method is really interesting. And they're thinking about ways that they can incorporate this new training method, which is really, it's like a teacher. It's like a personalized, it's a teacher coming in and going, I see, you're getting stuck on this corner here. Now, why do we go back to this corner and think about what it is about this corner that you're getting stuck on? And maybe we can back up and we'll move this way and maybe you can solve the problem this way. And so there is something about this training that is much more educational and helpful to the artificial intelligences. And someday it could be that curricula are designed and personalized by artificial intelligences for people.
I mean, I'm immediately reminded of going to school in the 90s and having the reading packets that were color coded based on your reading level, and you were assigned a reading level, and you would read a thing and you would take a little quiz, and then you would either get pushed up or you'd get pushed down. And so that was, a very 2D version of trying to personalize learning for a specific individual. And then you can adapt as you go with each new assignment or whatever. But that's just where my brain was thinking when you were talking about this, and thinking about having an AI be able to constantly adapt and augment your learning based on what you're getting and what you're not getting.
Right.
And that would be so fantastic, because that's what every teacher strives to do. But a lot of the time, if you have 35 brains in your class, it's really hard to do that effectively for all 35 at the exact same time.
Right. Yeah. And, teachers have different ways to address this, whether it's groups of students or having students work together, or, teachers do what they can. But yeah, sometimes, maybe if we had AI working on our online curricula to personalize them for people, maybe, I don't know, are AI teachers going to make sure more people don't slip through the cracks? Will they help teachers?
Yes, that's more what I mean.
We want them to assist teachers. Help the teachers.
Absolutely. So you can work on your reading packet like I was describing, right? But if it's AI assisted, then it might help hold your hand every step of the way in a way that an individual teacher cannot.
Yeah.
So it would augment their abilities, of course.
So it's okay. If you're a teacher because you want people to be educated and this works better than having a teacher, you should be happy to be replaced by a robot.
I don't think that. Oof. That's tough, because a huge part of being a teacher is socio-emotional. Huge, enormous.
Like, I don't think that AI are there yet for people being able to.
Depends on the teacher.
Yeah.
Learning. I guess my point is school is socio-emotional, right? And that can happen on the schoolyard, or in the classroom, or from your music teacher, or from your PE teacher. It can happen from it really should happen everywhere. But yes, that is the part that I think you're going to be really hard pressed to find an augmentation for.
I don't think so. I mean, I can see a lot of teachers who I would love to have replaced with AI over the years, especially as I'm going to tell a quick story. I felt really sick in school. This is the 4th grade, Quincy Elementary School, Quincy, California. Anyway, felt really sick. I told the teacher I need to go see the nurse. She said, "You're fine, sit down." And she wouldn't even felt. I'm like, "I have a fever." She felt my head. She's like, "You're fine, sit down." I got up and I went to the nurse's office.
Did you learn something about yourself and how to deal with difficult people?
No, I was just. So I went.
He learned that he's anti-authoritarian.
That's what I discovered.
He doesn't listen to teachers.
Well, even better. So I go to the nurse's office. I have 102 fever.
Whoa.
She's like, I'm gonna call and have you picked up from school. I'm like, okay, I'll be right back. I went back to the class. I don't know what. I'm like 8 years old. Opened the door to the class and yelled, 102! And then closed the door.
This tells me so much about you, Justin. 102.
102.
Oh my God.
Get out of here. But yeah, there's times when I think having a teacher there as somebody you can relate to and talk to, and just even if it's small talk, making you feel comfortable in the classroom amongst a bunch of strangers, peers, and other. I get what you're saying, that's an important factor to being teachers, but if it comes down to the education, and sometimes I felt like teachers got in the way of that, even the structure of the classroom sometimes got in the way of that. Yeah, absolutely.
It depends on the student. It depends on the classroom environment. It depends on the teacher.
And I grew up before they paid any attention to how anybody learned. Like, here's the material, here's my coursework, here's how it's being presented. You jump in line to the way I teach the class, and then you get something out of it, maybe not. And I think most teaching is still done that way.
Mm-hmm.
So.
Well, and meanwhile, I dealt with new math, which was a giant mess. So
the old math wasn't much better. I'll just say that. It wasn't easier.
And now it's time for me to talk about sleep. Make you sleepy.
Oh, how appropriate.
Yes, I know. I wanted to end the show on a couple of sleep stories that I thought were very interesting to have come out in the last week. 2 studies out this week showing the important nature of sleep. One published in Science Advances by a group out of Northwestern University examined fruit fly brain behavior, and were able to discover that while they sleep, there are fruit fly brain rhythms that are important to potentially get waste materials, toxic proteins, etc. out of the brain. And fruit flies are an important model for human neurodegenerative disease and other areas of study. The researchers they found that the fruit flies had proboscis extension sleep. This is a deep stage in sleep. If you ever wanted to know how fruit flies sleep, there is a stage in their sleep which is very similar to deep slow wave sleep, which we know is supposed to be very restorative for humans. During this stage, the fruit flies extend and retract their proboscis. So while they're sleeping, the fruit flies are like sticking out their little proboscis.
Are they dreaming?
Sucking it back in. Not during that stage of sleep, or I don't know if they're dreaming, but what they think is going on is that the pumping motion is moving fluids, and that is possibly triggering the motion of fluids through the little fruit fly brain and to the fruit fly version of the kidneys. And so they say that this PES, proboscis extension sleep, facilitates waste clearance and aids in injury recovery. When they impaired this phase of sleep, kept them from being able to stick their little proboscises in and out, they were less clear. They weren't able to clear an injected non-metabolizable dye from their systems and became more susceptible to traumatic injuries. So fruit flies, deep sleep, proboscis pump, and brain clearing. Additionally, another study that is from Penn State researchers and looked at REM and non-REM sleep in mice discovered, this is published in eLife, discovered that blood vessels in the brain become more dilated during REM sleep and then even more dilated in non-REM sleep than compared to when the mice are awake. And they think that this vasodilation of the blood capillaries, again, very important for flushing out the brain. Yeah. So sleep, it's important. It's like letting your brain take a bath. I'm just thinking about, I'm going to think about little fruit flies.
Well, isn't sleep also when your brain does a lot of the like clearing out long-term or short-term memories and either putting them in long-term storage or ditching them?
Yes.
So they're also doing that kind of like Marie Kondo cleaning of your memories as well.
Mm-hmm.
So it's a very cleansing ritual, sleep.
Sleep
I
is.
would recommend 10 out of 10 stars.
Would recommend. Yes. Report out to science. We have a letter, a question from a listener. This week in Science Questions. Hey guys, first, love the show. I started listening when you interviewed my friend Dan Hummer and have been hooked ever since. I'm curious if you can help point me in the right science direction. And shocker, it's a COVID question. How will we be able to evaluate if vaccinated people can be carriers for the virus or not? I'm thinking I'd like to propose this question when my spring medical physiology class starts in a week, but frankly, I'm not even sure, so any thoughts you have would be greatly appreciated. Thank you for all y'all do. Hope you are all healthy and as well as can be for pandemic times. Sarah.
My first instinct is that we need to have access to anonymized but tracked medical record data, because that's it. There's millions of people who have this, so it'll show up in a doctor's office on a test at some point. We just need access to be able to study it. Which sounds easy, but it's harder than you think, at least in the States, because of the privacy around medical records. Sometimes being able to track an individual's progression over time, as easy as that sounds, can be very difficult.
And then add to that, though, contact tracing. So if we implement more rigorous contact tracing and can determine who infected people, and if those records are, as you're saying, the anonymized vaccination data, or if people allow the fact that they've been vaccinated to be taken down on record during some of that contact tracing, the contact tracing interviews, then that could lead to it. But yeah, it would take hopefully people are trying to do this. I hope they're trying to get access to this data.
I've been doing a lot of research on this, obviously, because I have some very specific skin in the game, because my husband works in the ER and is vaccinated, but I am not. And so there's been all this conversation about the fact that the general understanding is we don't know. So we are assuming that if you are vaccinated, you can still spread. But so what I read was that the initial studies on the efficacy of the vaccines didn't really look at this question at all, because they were fast forwarding warp speed towards being able to deploy vaccines. Now they are looking at this question. And most research on this right now that I have seen is all about viral load. So just like you test the efficacy of a mask, a particular type of mask, by seeing how much virus gets from somebody's mouth into the air around you, they are looking at, if you get quote unquote infected, does the viral load increase, and are they shedding virus into the air around you? Because if COVID just gets into your body and you have the vaccine and it's like it's on a surface, right? Like it's not replicating, it's just sitting there. The likelihood of spreading that further from it being inside your body is pretty low. It's more of a question of, is it replicating in your body and you're just not showing symptoms? And so my understanding is it would take really intensive testing, and it would be longitudinal. They'd have to see if over time, is the viral load in your body increasing?
Yeah, I think I think that's another really, really important way to test it. The final possibility would be to do purposeful infections.
Mm-hmm. Right.
So
yeah, that's like the thing that
actually
we talk about, really.
Put vaccinated people together with unvaccinated people and see if and how many infections occur.
Yeah.
So you could actually.
Well, I mean, I'm a data point because everyone's using PPE as best they can, but there's still COVID positive patients in the ER where my husband works. He's wearing PPE, they're wearing PPE, but there's still, a possibility of exposure, and then he's coming home to me. So we could be a data point, but we'll see.
Yeah. And like you said, this was not part of the initial studies because they just wanted to see that the vaccines worked to protect people. But hopefully more of this data will come out as more vaccines are tested, as we have more people getting vaccinated. I mean, it's just, this stuff is a rolling process. And yeah.
Well, and if Justin's thing had worked, like if everyone had shut down properly, this wouldn't have been as relevant of a question, because people who had to be exposed because they had essential jobs could get vaccinated, and everyone else would stay the heck home. So it would all be much less relevant if the shutdown had happened more fully.
Yeah.
But yes, now we're having to ask this complicated question.
Now we do have to ask it. But thank you so much for writing in, Sara. It's wonderful to hear from you, and I hope that you have that you do ask this of your class. Anybody else out there who has a question, doesn't have to be about COVID, could be about any sciencey thing. Ask us a question at, where do you want to send it? Oh, to my Facebook account. This Week in Science is on Facebook. You can ask us there, or you can send me an email, Kirsten@thisweekinscience.com.
Yeah. It just, this was pointed out too, during the inauguration. There were people there, like Biden attended.
All vaccinated.
Who has been vaccinated and was wearing a mask. So maybe he's only had the first shot. I guess maybe there's a second shot to come, so there's not the full. Maybe that's why. But the other part of it is, they're maybe getting advice at that level that even though you're vaccinated, you still need to wear it so that you can't spread it. Or it could just be, hey, you have to set the right image. Can't really tell.
No, I mean, no, that's the current thing. They gave Brian a mask when they vaccinated him. They said, don't forget now, keep wearing your mask.
Yeah.
And he's had his second too. So they're still telling him, wear the mask so you can't catch and spread.
Mm-hmm. Correct.
Yep. Wear it. Mm-hmm. Send us an email. Ask us a question. We love to have questions from you. We've come to the end of the show. We've done it. We there? We're at the end. We have done it. I would love to thank you all for joining us for another episode of This Week in Science. I do hope that you enjoyed the show. Those dinosaur butt stories, they don't come that often, but when they do, we enjoy them. Shout outs to Fada for help with show notes and social media. I'd also love to thank Gord for help manning that chat room and keeping our chat room fantastic. Thank you to Identity4 for recording the show and keeping that going. Oh, I forgot to open my thing that I made from last week. Hold on. Here I go. I would also like to thank our Patreon sponsors and the Burroughs Welcome Fund for their support of TWIS. Thank you to dun 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 dun. Why isn't it sharing? It's not being nice. There. Oh no, my computer just decided. Am I still here?
Mm-hmm. Yeah, you're fine.
Okay, great. Then I'm gonna moved to this other computer, so at least we keep going. The thing that I tried to do did not work.
Pointing out that Barack hugged Lady Gaga. So there was an awkward moment.
Nope. Okay. Here we go. Thank you to Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Gaurav Sharma, Shubru, Sarah Forfar, Darwin Hannon, Donald E. Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Bentley the Translator, Big Nell, Kevin Reardon, Noodles Jack, Brian Carrington, Matt Bass, Joshua Fury, Shauna Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Melazon, Johnny Gridley, Kevin Railsback, Flying Out, Richard Porter, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood, 2020 can bite my shiny metal R's. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapoche, Sarah Chavis, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O. Kevin Parachan, Aaron Luthen, Steve DeBell, Rob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Gary S., Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts, and Dave Friedel. Thank you for all of your support. Woo, woo, woo! On Patreon. And if you would like to support us on Patreon, head over to twis.org and click that Patreon link. On next week's show, we're going to be talking with some scientists about mosquitoes. So if you want to know about mosquitoes, don't miss it.
Yeah. And when will we be talking to these scientists about mosquitoes? Wednesday, 8 p.m. Pacific time or 5 a.m. Thursday Central European time. You can also find us on our YouTube, Facebook channels, and from twis.org/live for the live stream.
Hey, do you want to listen to us as a podcast? Find out the things that got edited out. You can compare and contrast. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. You can also sign up for our newsletter.
Yeah. You can also contact us directly. You can email Kirsten at kirsten@thisweekinscience.com, Justin at TWISminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in that subject line, or your email will be spam filtered all the way into, let's say, a myelin sheath.
You could say that.
The dinosaur's cloaca, but
or a well-preserved dinosaur's cloaca.
That one, you see.
Also, you can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This week in science. This week in science. This week in science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
That is the end of the show, you know. It's the end of the show, the end of the show. My Google is not responding. Trying to share my screen, locked up my Google Chrome, and it's amazing that I'm still broadcasting. That I'm still streaming to you. That's good. But my Chrome is not responding, and I'm getting this blue spinning wheel.
Yarps.
Yeah. And I'm wondering if I just need to shut this window and if that will fix it. Which means that I will be gone for a couple of moments. Then I'll come back.
Okay. All right.
Can you hold the fort for 1 moment?
I'll hold the fort.
Okay. I'll be right back. It's all you. Maybe. Nope. Oh, I gotta just cut. Oh, I have to close the whole program. What a pain in the butt. Okay. I'll be right back.
Oh, okay.
Okay.
Okay. Yeah, let's see. I'm looking in the chat room. It was very cool to see so many living presidents in one place today. That was neat. I'm glad they were all vaccinated. Does make it a little less scary.
Back.
Oh, you're a third person.
Oh, look at me. I'm here again.
Yeah. Oh, there you are.
There. To remove myself and add myself. It's still down.
Okay. I need a clarification.
What?
On something that came up during the show. I've been still back in my mind, still trying to interpret something you said, Blair. And
Are you talking about the chocolates and candles?
I'm picturing they were using cloacas? What do you call them? Cloacas as molds for chocolates and candle holders. This is what my brain came up with. And
that was Blair's suggestion, but
that
is not.
May I screen share this?
No. No.
Okay, I'll just put it in. I'm putting it in the web chat.
You can put it in the chat. But no, you cannot share it on the screen.
It's just a piece of chocolate.
Just a piece of
Just
What are
a
you
piece of
talking
chocolate.
about?
I'm talking about using your own rear as a mold for chocolate.
That is what you were talking about.
Yeah.
Yep.
Okay, thank you for not sharing.
Are you in the web chat?
No.
Okay, let
And
me
I'm
put it
not
in the
going.
private chat.
I'm not going. It's
There
okay.
you go. It's in the private chat. But then also,
Yeah, because then it's worse. Then people are going to Google it on their own and find.
there's a whole nother thing.
Kids, people
Where-
do weird things in the world.
I'm going to use clinical terms
Oh,
here,
good
so I'm
God.
not trying to
good
be
God.
dirty here.
I have to unsee this.
But there is another thing.
Did he just hang up entirely? Did he just hang up entirely?
He might
Oh
have
my
hung up.
goodness.
That's very rude.
That's rude, Justin.
It's just a part of biology. Come on.
Is a part of
And then
biology.
there's a whole nother thing where there's a kit that you can buy to make a silicone mold to later make candles.
or chocolates.
Or chocolates, but it's often
it's a
used for
silicone-
candles.
yeah,
But you can make a
silicone mold.
silicone mold of your vagina. Is the other thing.
Yep.
So that is a thing you can buy,
Nope.
and then you can make. So I'm just
Candles.
saying, it would be neat to have a candle from a dinosaur butt fossil. I don't know. I think that'd be really cool.
See that would be cool. Yes.
That's what I'm saying.
No, Cheryl, not the goop thing.
No. Although that did remind me of this whole situation. But see,
Yes.
that was supposed to be a candle that smelled like.
Oh, Justin's back. He didn't leave
that.
entirely. I thought you completely hung up and weren't going to come back.
Okay, the candle that smells like what?
Gweneth Paltrow's nethers. So that's a thing that exists.
Why would anybody make a candle that smells like their butt? Okay.
No, not her butt.
No.
Oh.
Yeah.
No. I still think no.
Yeah, and then Cheryl, yes,
There's
and it blew up. Somebody
there's a-
bought one and it exploded in their apartment. So that's
Okay.
No.
Okay.
Yeah.
I have a couple of takes here, just real quick. First of all, who would buy that?
Who would what?
Oh, my
Buy
God.
that candle is one.
So there's a whole she has her thing called Goop, which is like her lifestyle blog, I think.
Okay.
And now it's also like an online store and like all this stuff. So it was part of that.
Okay.
I don't know.
Okay. Well, hey, people. Hey, Dr. Justin's not a real doctor. Poo pills, by the way, don't smell like poo. Just
Yeah,
so you know,
just-
just to make that clear.
All I'm
No
saying
other
is
tastes like.
don't yuck other people's yum if it doesn't hurt anybody, is my whole thing.
Okay, sure. Here's the other thing. You were mentioning the silicone molds.
Yes.
I've worked with silicone molding before. It generates a decent amount of heat.
No, I'm assuming so in order for you to make like for example, when you make a mold of a thing, you don't usually go straight to the original thing. You use like plaster of Paris. It's obviously not plaster of Paris. There's a whole kit. But like you'd use plaster of Paris to make an impression. Then you'd put like wax in that. Then you would use that to make a mold. So like there's like multi-step
Multiple
processes.
steps.
Yeah.
But so I have not researched it.
Yeah,
I do
don't
not
do
know
that.
how you go about making a silicone mold of your vagina. I don't know, but it's something that can be done. But this is what I'm saying is use that technology to make me a dinosaur cloaca candle, please. That's all I'm saying.
You could make all sorts of dinosaur fossil candles and other. I mean, that in itself is that idea. I like the idea. We went off on a tangent because of where the idea came from for you.
Yes, Stephen Rain in the chat room. New episode of How It's Made. Yeah, that's gonna be a good one. That's gonna be a real good one.
Nope.
Yes, I must admit, I am not well versed on any of it.
I
just know it exists.
Yes.
Oh, you know what I could make as part of my lifestyle brand? Huh? No.
I think what you're thinking about, I'm going to tell you right now,
No.
also exists.
Yeah, it does. I'm sure.
It certainly does.
Yeah, this will be Hot Rod. This will not. I mean, maybe dinosaur fossils coming to a TWIS store near you soon.
Yeah.
But none of the no other stuff.
No.
I want.
We're a science show, people.
Seriously considered buying a life-size T-Rex skull replica.
That would be really cool.
You said the word life size, and I got very scared based on our conversation, and then it ended great.
It ended wonderfully.
Here
Yes.
you go.
Okay. It was, I think, 10 grand. I think it went down to like 6 grand, something like this. But it has like a whole mounting kit and everything, and it's huge. It's a full size.
Is that the one that Nick Cage owned? Because I know that he was auctioning off a T-Rex skull. When
maybe.
he was like auctioning off all of his personal possessions. Remember that, like 10 years ago or something?
He ran out of money because he made poor choices.
Oh.
Poor Nick Cage. Oh, and now he's doing really hilarious stuff.
Is he?
On television and movies. Yes. Oh, I gotta find what his.
There's the
most
thing
recent stuff is.
he did a bit on the use of curse words.
Yes.
The origin of curse words.
The origin of curse
which
words.
I think it's hilarious because he spends this whole bit using the F word in different intonations to express all the different meanings you can get from that single word. But what's hilarious about him being the one that does this is one of my favorite things about him is he got the script for The Rock, which is this him and John Connery,
of all time.
and they go to Alcatraz, and there's this whole
there's a bioweapon. Yeah.
Yeah, he's like he's not even an agent, but he's the one that's got the expertise.
Stanley Goodspeed.
But he had, I think, he was like, yeah, my kids were just at the age where they're going to be watching my movies, and he went through the script and edited out all of the curse words and came up with other things to say throughout the whole script. And if you know that going in, it sounds like the TV version where they've thrown in like the oh gosh when you're supposed to, like, when stuff's blowing up instead of the curse word that was said.
That's funny that Michael Bay allowed that.
And he did it. And he was the biggest, one of the biggest stars at the time. And so he got away with getting this done in the script, like you said. And it's, if you know it going in, you pay attention to it in that movie, it's hilarious. It's just another element that makes that movie. It's, I think, the only thing I really liked about that movie, as far as I can remember. But it was really hilarious.
Versions. I love that movie.
There are versions you can get of a lot of movies that have been edited that way for kids, where
right. It was the TV version.
It's like family. It's like edited for families, and it's but that it's on Netflix. The History of Swear Words or something like that, and it's great. The one I want to see is Jiu-Jitsu, because I love bad kung fu movies, but this one's Jiu-Jitsu, not kung fu, and the trailer looks awful and wonderful at the same time. It's going to be amazing. You need to see the trailer. You do. It's so wonderfully good bad.
Although we're going to talk just for a moment about the martial arts as physical combat. Has the martial arts sort of been delisted? Because you got this with the UFC, the cage match fighting techniques, and it always seems like it's these weird street fighters that just come out and punch people that win these things. And the guys with all like the cool moves and the stuff and the doing the kung fu jiu-jitsu thing eventually just get taken out by somebody who punches people really hard. I don't know. I think if you look at the what's going on in actual hand-to-hand combat competition-y stuff these days, there's techniques that come from all of these that people learn, but ultimately seems to come down to somebody who's better at punching somebody in the face.
You talking about the cage match stuff?
Yeah.
That's different. The ultimate fighting. Yeah.
Whatever the thing is called. It's different. Usually like one of the couple of the least ones that I've
Joe Rogan hosted things
have been like the champions were like Miami or Irish street bar fighters. like just backyard brawlers who somehow became the best fighters against all these people with all these medals for competitions and doing these circuits. They were like, I'm a triple, quadruple black belt, and this guy grew up in a rough part of Scotland. Bam! The guy with the rough part of Scotland punches him hard, and the other guy falls down. It's I don't know. Which is, it kind of reminds me, I had this this friend in high school who had been studying all of the martial arts. He had like the fast punches. He could show you different ways he could throw you to the ground really quickly. And it was like always very impressive. And then he took his first wrestling class, thinking all of his training was going to come and he'd be good at wrestling. And he just got beat right away and badly all that. And he got really into it. He became a wrestler. He's like, this is where it's at. This is where it's at. Because nothing that I have learned in all of my years of taking all of these classes really prepared me for somebody grabbing you and bringing you to the ground. Like
that was the Gracies.
I had nothing to do.
That was the Gracies who started for the mixed martial arts. Yes, the MMA stuff. Marked. It's the Gracies were doing jiu-jitsu, Brazilian jiu-jitsu, and they were doing the grappling stuff. And the Gracies they combined a bunch of their jiu-jitsu with kickboxers and boxers, and so like people started mixing all these things together. But I think it was the Gracies that really got it started. And it's fierce. It's brutal.
Yeah, I
like watching the fights every once in a while.
Oh yeah, well you've got like a black belt in something though. You're like a is it Kempo?
Taekwondo.
Thank you.
Yes. The way of the fist. Taekwondo. It basically means I'm going to punch you now. No.
And I say this, and then now the people who have been trained in martial arts will come up to me now wanting to prove that their fighting style works on me. Any fighting style will work on me. Just so you know, it's not a high bar. Kicking my ass is not a high bar for your ability to fight. It's just won't be.
You gotta stretch out a little bit. I can kick and punch and kick.
Yeah.
I mean, if you're on the street, though, Justin, the thing is you can always just run away. The cage match, you're kind of stuck.
You're stuck in a cage.
Yeah. I actually did learn two pieces of martial arts that I have found effective. They were both from Kempo Karate. One was a block with the hand. Another was a block with the knee. And I found if you ran at somebody and you did both of them, you could get by them and keep going.
Ooh, yes.
Yeah. Very, very handy technique.
Very useful, yes.
Yeah, track is actually my martial art.
Being able to run. I love, what is it? Aikido. Aikido is, it's supposed to be the very, energy redirecting martial art. It was Jean-Claude Van Damme's martial art.
No. Oh, was it Jean-Claude's? Okay.
Yeah.
But he
Black belt
did... I always get him confused with the actual martial arts of Frank Dux, which was the ninjutsu.
Yeah.
Because of the blood sport kumite.
It's fun stuff.
I did take a kendo. Was that the one with the hit people with sticks?
Kempo.
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 But we didn't get to use actual swords. We used these wooden sticks and like the thinnest little hat thing. But from what I could tell, the class was basically to give the more advanced students people to beat up with sticks.
Right.
I'm like, I'm done. I'm not. Am I paying money to get concussed? I'm out of here.
Oh, Seagal. Thank you, Ananuki. Steven Seagal was Aikido. That's what it was. Yes. That's the those two fighters. Both of whom I think ended up with like drug problems and
Oh. Well, like Jackie Chan.
Jackie Chan is amazing.
Also claims to have broken every single bone in his body.
If you've seen the outtakes from all the movies that he's been in, I can believe it.
Yeah.
There are some movies where you watch the outtakes and then like you realize that at some point halfway through the movie he did a stunt, broke his ankle and was in a cast for like the rest of the filming, and you're like, wait a minute.
He does have my favorite martial art movie scene ever. I think it's called The Brawl, the movie. It might be wrong. But his grandfather has told him he is not allowed to fight. And so when the thugs come to start stuff with him, he's sweeping up the shop and accidentally hitting somebody, and then he's doing the dishes, he's moving that. He's like he's continuing to work.
I'm not getting in a fight. I'm just doing my work.
Yeah, it's got this level of slapstick to it. That's fantastic.
He's brilliant. I think he is a massive star in China and I think he's one of the richest. He's among the richest people in China, I think I heard somewhere.
That might have been true in the late 80s.
Yeah.
But I think that
no, because he's a producer. He's a producer, a director, and a singer, and he's like this. He's sold so many records in China. Yeah. Hold on here.
Seagal lives in Russia now? Is that true? Oh, okay. All right.
Seagal is in Russia.
He did always.
Okay, maybe not.
He did always have like this weird Russian entourage thing going on. I heard about this. This is a story on LA. He had like this weird Russian mob entourage thing going on all the time.
Okay, so actors in the world. He's in the top 20 richest actors in the world. Jackie Chan is.
There we go.
He's somewhere around $400 million.
I think most of the world's billionaires.
And he deserves it.
Yes.
There's
hanging off of the side of buildings and stuff.
And he's just brilliant, and he seems like he's just this nice guy. Seagal.
He's a seagal.
Kendo. Kendo is the sword fighting. Kempo is the sticks. There's a Sadie.
No, Kempo is a karate. Kendo is the sword fighting. Kempo is a karate style. That's the one I learned.
Karate. Okay.
I learned a little bit of Kempo. That's where I got the blockity blocks. Yeah, Kendo is the concuss new students until they get a thick skull or leave.
Mom, let's go to bed.
Aww. I would like to get back into martial arts. I have not done martial arts in a long time. It is fun.
You have enough martials going on right now.
I have a martial. I have a marriage in the martial arts. That's a cute little piece of hair there.
Yeah, I got like an alfalfa thing going on.
Mm-hmm. You do. It's fantastic. Kendo swords. Thank you. Should we head out for the night? You know, I'm just very happy to have had a very nice day that didn't seem to have violence.
No.
When it was expected in places, and there's a lot of.
Do you know why you didn't have any violence? Because it was expected in places.
Yeah. Yeah. But I'm very today was a good day. Let's have more good days. I would like to have more good days. Let's go to sleep so we can have a good day tomorrow.
Yeah, so I can wash my brain.
Mm-hmm.
And I might not see y'all next week.
Yeah, so you're potentially traveling.
I'll be traveling that day, I believe. So
Okay. So no mosquitoes for you.
I got it.
Let me know if you have any questions about mosquitoes. You have a week to think about it.
Okay. I don't know. How is it going to be about? I do have some questions about it. I do want to know how much of a pollinator they actually are. Because
I had a feeling you were
I
going
don't
to
want to get rid of them,
Yeah.
but I will take the advice if they are a good pollinator to put up with them.
I think that is a very, we want to know that.
Yeah.
Things we want to know about mosquitoes. And I'll tell you more. We'll have more next week. Identity 4, I was contemplating the answer to your question, which is why I hadn't replied yet. I think if you sent the audio I don't think that is useful in the moment. I think just sending the full file is best for me right now. I think if we start doing something, I got to think on it a bit more. I think I need to do more intros and outros that we record with each episode that would be then able to be cut in and cut out if we decided to cut shorter bits for the show. But yes. So I'll
And do we need us?
What you're doing is great. Thank you.
So I see a lot of shows that don't have intros for their shorts. They just start right with content for the short version. And then at the very end, they tag what the show is. Here's the listen more. There's a...
Is that on YouTube or for like podcasty stuff?
I'm talking about YouTube, not podcast.
Yeah, okay.
Because I don't really listen to those. I just kind of have a lot of stuff on YouTube. I track.
So if we just like Jimmy Fallon type stuff,
Well, I was thinking,
or the clip and then.
Yeah, do the clip and at the end, hey, you've been watching this show, blah, blah, blah. For more content, subscribe, hit the like button, blah, 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 right at the end.
Yeah.
Which is sort of a given now anyway, that if you liked something, you would do it. So that might even be a little redundant at some point. But
yeah. Ooh, Identity 4 has a back pocket process if we want to get fancy. I like fancy, I think. Fancy or not fancy? This week in fancy. Now, I fancy saying... Good morning, Justin.
Good morning, Justin. Say goodnight, Blair.
Good night, Blair.
Good night, Kiki.
Good night, everyone. Thank you for joining us on this hopeful eve. And I do hope that you have a wonderful week. Stay safe, stay healthy, and fill the world with science and kindness. Kindness, compassion. And we'll see you again next week. Take care, everyone. Grr, grr. Be kind, Sadie. Be kind.